Hello everyone, welcome one more time to the science class of class 10. We are going to cover today the remaining portion of digestive system. So I'll try to keep my class a little shorter than the last time, mainly because very long videos are difficult for people to watch later on. So I'll try to go a little bit faster, but if you have any difficulty understanding me, I will clarify your doubts in the chat or on my WhatsApp. So you can note down my WhatsApp number 721-8199-666. So if the clarity is okay, you can write down on your chat window, it is clear, and I can start with my session straight away. So is it clear? Namaste everyone. Yeah, Harsh Kumbhare wrote down my mobile number properly. So let me just check if I get the right answer from you, that it's clear, and I'm waiting for your green signal. So if the picture is clear, the slide is clear, wonderful. So most of you have written it's clear, and I'll come to this point. So what I told, remember, I will go a little bit fast, and if you have problems, you can ping me on my WhatsApp number after the class is over. That is after 2 p.m. because I have another class at 1 p.m. So let's come to the point and start with the presentation. So we have covered certain slides. I don't want to show those slides all over again. Let me just check if I can start in form of a slideshow from beginning and if it's visible. The moment I see that slide on my mobile screen, I can understand that you're able to see it, and then I'll go to the next portion. We have covered all these things: composition of bile and emulsification. This example I gave you, this diagram I showed you last day, if you remember. What is pancreatic lipase? That also I told you. So all these things I've covered. Pancreatic juice properties I've covered. So trypsin, chymotrypsin, how trypsin is activated by the help of enteropeptidases or enterokinase, that we have covered. I think the screen is getting stuck if I go for the full screen, so I'll not go to the full screen, I'll keep it this way. So pancreatic amylase, we have covered that as well. Intestinal juice, and these things are covered already. Now we will go to the next slide. The next slide, we will talk today about brush border enzymes. Now, have you heard this term before? Brush border enzyme. So brush border enzyme, what are these things? I told I will not be too slow today because we have got a lot of things to catch up. So what are brush border enzymes? Brush border enzymes are the most, I'll show you with a comb, maybe it will help. So in the intestinal lumen, inside the intestinal lumen, there are these structures I showed you last day. Remember, these are called villi. Villi, the finger-like projections into the intestinal lumen for absorption are called villi. Now a villi, if this is one villi and it is like this, it has got other microvilli attached to it. The microvilli is attached to the villi, and these are called brush borders. Why are they called brush borders? Because it looks like a brush. If you have seen a shoe polish brush, the shoe polish brush handle is the villi, and from it, just like different hairs are protruding out, these are called brushes or microvilli. So microvilli, what is the importance of microvilli? The importance of microvilli is it increases the surface area of absorption. खेल, remember one thing, ये पूरा खेल जो है digestive system का surface area का खेल है, because the more surface area you get, or optimum surface area that you get, it should be matching with your type of food that you take. जिस तरह का खाना आप खाते हो उस तरह का डाइजेस्टिव ट्रैक्ट का लेंथ और उसका सरफेस एरिया अगर नहीं हुआ, in that case digestion will not work properly. So understand one thing, our digestive tract, or for that matter any digestive tract, is depending on the surface area. So these brush border that we are talking about increases the surface area manifold. It increases manifold the surface area. So that is very, very, very important if you understand this. You see the carnivorous animals like lions and tigers, what happens to them? What happens to them is they have a shorter digestive tract. It is not as long. So what happens? The reason is they are putting in flesh, and flesh, if it is put in, it does not require so much traveling, because you're not trying to digest something that is difficult to digest. Proteins are there which are not so difficult to digest, but animals which have got certain vegetable components that they eat is not digested properly. The vegetable components that we eat are not digested so properly. So a vegetarian or a very simple herbivorous animal needs a longer digestive tract, and the villi is helping to making the digestive tract even longer by actually increasing the surface area. So brush border enzymes, the most important brush border enzyme is dextrinase and glucoamylase, which further break down oligosaccharides. Now what are oligosaccharides? Oligosaccharides are two chains of glucose attached with each other. Remember we covered last day that things like lactose or sucrose, these are all oligosaccharides. So dextrinase, I'll talk more about dextrinase later on. So these are important brush border enzymes. Other brush border enzymes, why are they called brush border? Because this is the brush and it is located on the border of these things. If you can see the picture clearly, I'll try to increase the size of the picture. See, this maltase that I'm pointing at right now, it's located at the tip of the brush. These are the microvilli. These are the microvilli. One single is called a villus, microvillus. So the maltase is located on the tip of the brush border, and so it is called a brush border enzyme. So keep that in mind. Brush border enzymes important are dextrinase, glucoamylase. Other brush border enzymes like maltase, sucrase and lactase we covered last day. If you remember, maltase brings glucose. Lactose is getting converted to glucose and galactose by lactase. Sucrose is getting converted by sucrase to glucose and fructose. So these are all brush border enzymes, and it is located on the microvilli, the tip of the microvilli. So keep this in mind. So lactase is absent in most adult humans, and for them lactose, like most polysaccharides, is not digested in the small intestine. So only the babies are meant to have milk, and that is the reason the milk digesting enzymes are not very prominent when the person or the individual grows up into adults. So keep that in mind, that milk digesting enzymes are mainly present when the individual is a child or an infant. Now we'll come to the next point. Digestion in mouth. Very quickly, I'll cover the biochemistry part of it. Digestion of carbohydrates starts at the mouth. In mouth, food undergoes mastication. Mastication is the art of chewing. During mastication, food comes in contact with saliva secreted by the salivary gland. Saliva contains what? You know that, ptyalin. Action of salivary amylase. This is very important now. If you do not have a pen and paper, bring it and quickly note it down. It requires the chloride ions for activation of and keeping a pH of 6.7. So pH of 6.7 that is there in the mouth, it requires the chloride ions to do it. The enzyme hydrolyzes alpha-1,4 glycosidic bonds at random deep inside polysaccharide, starch or glycogen, producing dextrins, maltose, maltotriose and glucose. I'll very quickly show what happens in the mouth. Amylase, this is a glycogen molecule or a complex sugar, and there are these bonds, alpha-1,4 bonds. 1,4 means under this ribose chain, what's happening? One is located at the site number 1. If you remember the hexagon, 1 point and 4 points, these 2 points are matching. That means location number 1 and a location number 4, they are getting attached to each other to form a bond, and that is called alpha 1,4 bond. And alpha 1,6 bond is the one side, the vertex number 1 and the vertex number 6 of another sugar is attached to each other, and then this is broken by the amylase and it forms maltotriose, maltose and isomaltose. Next, so intestinal brush border enzymes. Intestinal brush border enzymes are maltose broken down by maltase into glucose and glucose. Isomaltose by the enzyme isomaltase is broken into glucose and glucose. That is 2 molecules of glucose. Sucrose under the action of sucrase, which is again a brush border enzyme, breaks down into glucose and fructose. Lactase is again lactose is broken down by lactase by glucose and galactose. So lactose forms glucose and galactose under the action of lactase. And limit dextrin. Now you must be wondering, what is a limit dextrin? So if you want to take a note, you make a note of this. I will define 2 things, very quickly note it down. Dextrins are a group. Are you ready? Are you ready? If you're ready, let me know. I'll dictate and then you can make a note of it. If you're ready, be ready and let me know you're ready with a pen and paper. I will dictate dextrins and limit dextrins. Explain in short, very quick. Great, you are ready. So just very quickly note down, dextrins are a group of low molecular weight carbohydrates. Dextrins are a group of low molecular weight carbohydrates produced by the hydrolysis of starch or glycogen. Just keep it simple. We do not need to go deep into it. So it's a low molecular weight carbohydrate produced by hydrolysis of a starch or glycogen. And limit dextrin, the limit dextrin is a name given to the highly branched core of the polysaccharide fragments remaining at the end of exhaustive hydrolysis of glycogen. So I'll repeat again, limit dextrin is the name given to the highly branched core of the polysaccharide fragments remaining at the end of exhaustive hydrolysis of glycogen. So if you have noted it down, fine. If you have not noted it down, you can get back to this video again and watch it and you will get it. So now these are the intestinal brush border enzymes that breaks down sugar. Now macromolecules to micromolecules. This is the real journey, you know. This is the real journey that we are going to learn about absorption. Now you see there are 4 types of macromolecules that are there. There are 4 types of macromolecules that are there. And before I start going deep into the macromolecules, I'll just give you a 30 seconds breather, a sort of a short break, so that you can, think if you have understood properly or not. 30 seconds breather. Any question, you can ask me right now. If you have any question till the time I have covered, you can ask me right now. All right, fair enough, 30 seconds done. I'm going to the next point. So there are 4 macromolecules. You know that, right? One is the protein. The second is the carbohydrate. We can call it carb in short form. The third is nucleotides, which makes up the nucleic acids, DNA. The 3 major parts we will discuss later on. So first is protein, the second is carb, the third is nucleotides, and the number 4 is, yes, right, fats. Now what happens to these things when they undergo digestion? So proteins are amino acids attached by peptide bonds. So amino acid attached by peptide bonds. So to break these bonds, I've used that blue color over here. If you see peptidase, so to break the peptide bond. Now you know everything that is used for breaking, every enzyme used to break something, at the end we use ASE. So peptidase is used to break the peptide bonds of proteins and form amino acid. Now at the same time, the pancreas secretes 2 enzymes. What are these? Trypsinogen and chymotrypsinogen, or chymotrypsinogen. I'm spelling it deliberately wrong so that you can remember the spelling. So trypsinogen and chymotrypsinogen, under the action of enterokinase or enteropeptidase, they form trypsin and chymotrypsin, which in turn breaks down the peptide bonds. So amino acids are proteins which has peptide bonds. Peptidase is the type of enzyme that breaks down the peptide bonds and forms into amino acids. Now the pancreas secretes trypsinogen and chymotrypsinogen. These type of enzymes which has a gen at the end, GEN, these are called zymogens. Zymogen basically means whenever you see the word gen, you can understand that this is not ready for action yet. It requires one more push to get into action. So whenever the zymogens are there, trypsinogen, chymotrypsinogen, under the action of enteropeptidase or enterokinase, they form into the next, the active enzyme called trypsin and chymotrypsin, which in turn breaks down the protein peptide bonds and peptidases and starts functioning. Now carbs are simple sugars attached to each other, and these carbs are broken down into the simple form of sugar, that is glucose or galactose, by the action of amylase. Whenever you hear the word amyl, you understand it's talking about sugar. Amylase, something that breaks sugars. So simple sugars attached to each other are broken down by the action of amylase. Now nucleotides, how does the nucleotide look like? They are broken down into nucleosides. Now I'll show this picture to you one more time. This is a blown up picture. Now you see this is a nucleotide that forms with a phosphate group attached with a ribose sugar and a base. So this base is, if you remember the terms I told you, adenine, guanine, all these A, U, G, C, T, these are the things that I told you last day, if you remember. So that is, this blue part is exactly that. So the phosphate part attached to the ribose sugar and the base which is there. So when this is broken down, the nucleotide is broken down, it forms the nucleoside. Now what happens when the nucleoside is formed? You see the phosphate group is separated and the base and the sugar is remaining. So the base and the sugar is remaining. So that is the difference between nucleotide and nucleoside. So when these nucleotides are broken, the 3 major parts, the phosphate group is separated and the enzyme is called nucleosidase, not nucleotidase, because it is forming a nucleoside. So nucleosidase is the enzyme which breaks down nucleotides and separates the phosphate group. And the third part is the fat. The fat has got 3 parts as well. The triglycerol head that has an oxygen group on it, and then each oxygen has a fatty acid tail. So the triglycerol and the fatty acid tail and the oxygen group, these are to be separated as well. So we'll go to the next slide. Now see, we are talking about the villi right now. A singular is called a villus. So when we talk about villus, you have to see this is a 0.5 to 1.6 micrometer. So it's very, very small. So villi are singular, villus are small finger-like structures in the small intestine they help to absorb digested food. Each villus has microvilli, which increases the surface area of the intestinal walls. We just learned about the microvilli. Remember the comb, microvilli, and this is the villi. So it increases the surface area of the intestinal walls. A large surface area allows nutrient to be taken in more quickly. So when there's a large surface area, the nutrients can be absorbed very quickly. Now in the villi, how the absorption happens. In villi, monosaccharides, peptides and amino acids are absorbed either by diffusion or by active transport into the blood capillaries. Now if you see this picture, I'm again going to make it bigger. You see this red capillary that you see over here. Sorry, I moved the picture around. Just fit it in. Yes. So this red capillary that you see, this is a blood capillary, and you see there is something in violet or purple, and this is called the lacteal. So what is a lacteal? What is a lacteal? If anyone knows, you can answer me right now. What is a lacteal? If this red one is a capillary, what is a lacteal made up of? Or what exactly is a lacteal? So I'm waiting for the answer. What is the lacteal? Who will let me know first? Yeah, these are, Parth has answered rightly, Som has answered rightly. These are all lymph vessels. And you can see over here, which will come handy later on, that in the lacteal, the size of the lacteal is more wider than the capillaries. Now it has a very important role to play. I will come and tell you later on when we reach the point. So just see this picture and understand the lacteals are wider than the capillaries. Now the absorption. So amino acids are absorbed by primary active transport. Now I told you, remember the last day when we were covering these topics, what is primary active transport and what is secondary active transport. So primary active transport, if you know the answer, you can share with me. What is primary active transport? What is required? What is required for primary active transport? The picture I'll not zoom right now. I'll zoom when it is needed. You answer me whatever I'm asking right now, so that we are in touch with each other in a proper way. So what is a primary active transport? What is required? What is the raw material for that? Very good. Amay gave the right answer, ATP. The adenosine triphosphate is required. So what happens? The adenosine triphosphate loses its phosphate bond. In the process, the energy that is liberated is allowed to push a particle inside. The amino acids will be pushed inside the enterocytes, the cells of the intestine. So this is the intestinal lumen. These are the cells. So if I take a part of this, so intestinal lumen to inside the cell, it goes in. So it will go inside a cell. So if this is the cell of the intestine, a protein particle will enter the cell of the intestine only by the help of active transport. That is when ATP is used. Now comes the most interesting thing. What happens during secondary active transport? Who is responsible for secondary active transport? I want the answer for this. Who can tell? Let me see. What happens during secondary active transport? What is needed? Very quickly, I said I'll go a bit faster today. It's already 12:34. I'll wind up by 12:55 max. So who will be responsible for secondary active transport? Because the monosaccharides, that is the glucose, the galactose, these are pushed inside the enterocytes. These are absorbed by the intestinal cells called enterocyte. Entero stands for intestine, cyte stands for cell. So enterocytes absorbs these things, the amino acids, with the help of primary active transport, and the secondary active transport helps monosaccharides to be pushed in. No, here proteins are not required. Here we need something else. Now I will blow up the picture which you wanted. The picture is zoomed in right now. Now you see this is high sodium and this is low sodium. So high sodium to low sodium, what does it mean? High sodium to low sodium, it means that from the intestinal lumen, sodium enters the enterocyte. Sodium enters the enterocyte, that is the cell of the intestinal lumen. So from the intestinal lumen, it will enter into the cell of the intestine for absorbing, and when this happens, sodium moves from a higher potential to a lower potential. So when sodium moves in from the intestinal lumen, inside the intestinal cells, it moves from a higher potential to a lower potential, and that movement from a higher potential to lower potential liberates some amount of energy, or energy is released, and that helps the sugar to be taken inside the cells as well. So understand one thing, the sugar also comes out of this intestinal lumen and gets inside the enterocyte along with the sodium, which is actually deriving the energy by moving from a higher potential area, that is area of higher concentration inside the lumen, to the inside the cell. So I hope I have made myself clear. So sodium ions are responsible for secondary active transport by moving down from a higher potential to a lower potential. Now nucleoside, again active primary transport, or sorry, primary active transport rather, where it happens what? The ATP is used for getting the energy. But fats is a different story. So are you clear with all these things? Amino acids, how it will be absorbed? By primary active transport. Monosaccharide, how it will be absorbed? By secondary active transport. And nucleoside, it will be absorbed by primary active transport. So if you are clear with all these three food particles, the macromolecules, amino acids, monosaccharides and nucleosides, you let me know that you are okay till that part, because fat is a different story, and then we will go into the story of fats. Amma has written, sir, but the amino acids are absorbed passively by diffusion, and only L amino acids are absorbed by active transport. And inside it's written the amino acids are absorbed by active transport. Yeah, Amaya, you know the proteins are actually absorbed by active transport as well. So proteins are, amino acids are, if you go into the higher books, the books of medical stream, or if you even go to Khan Academy, you will be seeing that it's primary active transport which is required to push in the protein. Diffusion, yes, at times diffusion do happen as well with certain proteins, but not with everything. So D amino acids and L amino acids, you have written that very correctly, Omiya. So both these things are different, and it needs active transport to be pushed in. So now amino acids, monosaccharides and nucleosides, these three are clear. Now we'll go to the story of fats. Now you see, before we go to the fats, how the villi looks like. The blood capillaries absorb glucose, amino acids and vitamins and minerals, some fatty acids and glycerol. So these are the things that are absorbed by the blood capillaries, and as I said, by active transport and by diffusion. Now when I say active transport, I do not rule out diffusion. It requires diffusion as well, and it can happen through active transport as well. It depends on the nature of the glucose, or the sugar rather, and the nature of the protein. So if the molecules require some push, it is there to be given by the active transport. And in the lymph vessels, now why it cannot absorb the fats? Why these capillaries cannot absorb the fat? Mainly because the fat molecules are bigger in size. Now you must be wondering, I just told the macromolecules are broken down into micromolecules, then how come it becomes bigger again? So that story we will listen when we go into the fat story. So lymph vessels, lacteals, they absorb oil droplets, fatty acids and glycerol. because they are wider and macromolecules can go inside them. Now, how did this fat become a macromolecule again? So let's see the journey of fats. We'll go for a reading of this and then I'll explain. Triglycerides are emulsified by bile and hydrolyzed by the enzyme lipase, resulting in a mixture of fatty acids, di and monoglycerides. So remember, under the action of bile and lipase, that is pancreas gives the lipase, the fatty acids, like the fat is broken down into fatty acid. The bile does the hydrolysis, in fact, emulsification done by the biles and the lipase causing them to break the chain of fatty acids, and then it is broken down into smaller particles. But these then pass from the intestinal lumen into the enterocyte. So the fat particles, the fatty acids, glycerol, these move from the intestinal lumen into the enterocyte or cell of the intestine, where they are re-esterified to form triglyceride. So again, they are esterified. So what is ester? Ester is a chemical compound derived from an acid, normally organic or inorganic, in which at least one hydroxyl group is replaced by an alkyl or alkoxyl group. Alkoxy group, okay? So one alkyl group is replaced by one hydroxyl group is replaced by O-alkyl group. Do not get confused about it. It's higher science, but remember one thing, ester is a chemical compound derived from an acid. So let's get back to the point again where we were covering. They are re-esterified to form triglyceride. So the fatty acid inside the intestinal cells, again they join together to form the triglyceride. The triglyceride then combine with phospholipids, cholesterol ester and apolipoproteins, which is a mixture of lipid and proteins, in B48 to form something called chylomicrons. So the triglyceride then combine with phospholipids, cholesterol ester and apolipoproteins to form something called chylomicrons. So this term you remember, chylomicron. So when it goes inside the fat, broken down by the action of bile and lipase, gets inside the intestinal wall, and inside the intestinal wall they again join together and they form something called chylomicrons. These chylomicrons then pass into the lacteals, forming a milky substance known as chyle. So then these things go into the the lacteal, the lymph vessels, and it is now known as the chyle. So when in the stomach it was known as the chyme or chyme, and when in the intestine it is known as the chyle. Chyle is nothing but the fat forming again the triglycerides, which gets inside the lumen. From the lumen inside the intestinal wall, they form again something called chylomicrons, and this chylomicron is known as the chyle. The lacteals merge to form larger lymphatic vessels that transport the chyle to the thoracic duct, where it is emptied into the bloodstream at the subclavian vein. So the chyles now goes from the lacteal to the larger lymphatic vessels and transported to the thoracic duct, where it is emptied into the bloodstream. So again, it goes into the bloodstream at the subclavian vein. Now come to the next point. I don't want to confuse you too much. This part is a little bit difficult, but understand, पहले फैट था, उसको तोड़ा, ट्राइग्लिसराइड, fatty acid का chains अलग अलग हो गया. Understand, the amino acids, the carbs and the nucleosides, they are absorbed into the bloodstream directly, either with the help of diffusion or with the help of primary or secondary active transport. So either of these mechanisms are responsible, or both of these mechanisms are responsible for pulling in carbohydrates, amino acids and nucleotides. But when it comes, not nucleotide, it's a smaller one, nucleoside, the broken one, the phosphate group is separated now. But when it comes to fat, the fat is broken down under the action of bile, which emulsifies it into fatty acids, and this fatty acid is broken down with the help of lipase enzyme secreted by the pancreas. Now when it goes it's broken down, it's absorbed inside the villi, inside the villi, and when it enters the villi, they are again clumping together to form what is called the triglycerides, and these triglycerides are moved from the lacteals, and from the lacteals it goes into the blood flow. Now I think there's a problem with the internet connectivity. Are you able to hear me and see me properly? All right, I think the net connectivity is a bit slow. Let's come to the next point very quickly. Liver. So liver is called the coolest organ. So liver is called the coolest organ. The reason is it's responsible for metabolism, that is catabolism and anabolism. The storage, the carbs and the fats are stored in form of glycogen and lipoproteins and triglycerides. So remember one thing, the liver stores the carbs and the fat by converting into glycogen. The proteins are not stored in liver. The proteins are processed into molecules like albumin, and the liver processes it. The protein is converted into albumin, and that albumin is cast off into the bloodstream, where they float around and carry on other functions. So liver क्या करता है? प्रोटीन को स्टोर नहीं करता है. It converts it into albumin and it goes into the bloodstream, where they float around and they carry on other functions, unless the liver needs to again pull it back, all the albumin, and convert it into other molecules for different use. And liver also does something very, very important, that is called detoxification. So cytochrome P450 enzyme is one enzyme which is very, very important for detoxification, because it reduces the intensity of the toxin. And so when a medicine is taken inside, the medicine is also, the power of the medicine is reduced by this cytochrome P450 enzyme. This P450 enzyme, cytochrome, is reducing the efficacy of the medicine, and that is what the doctors do. They calculate in their mind what portion will be actually acted upon by the liver cytochrome P450 enzyme, and they give the dosage accordingly. So this is very, very important. Now you come to the next thing. Why liver is important? The liver has got two separate blood supplies. The liver has got two different blood supplies. The first one is a portal vein. The portal vein, it comes from the intestine or intestinal tract and supply nutrient-rich blood to the liver. So if you see over here, the portal vein, this one that I'm moving the thing with, my mouse with, is the portal vein. The gastrointestinal tract, the hepatic portal vein, brings nutrient-rich blood to the liver. What are these nutrients? What are these nutrients? These nutrients are nothing but the ones that was absorbed directly by the blood, or the capillaries, that is sugars, the carbs, and the nucleosides. The second thing is it has a proper hepatic artery. That means who will supply oxygen to the liver, because the hepatic portal vein is only supplying the nutrients. The hepatic artery, it comes from the body to the liver. From the heart, it brings oxygenated blood to the liver to carry out its functions. And The third thing is there's a hepatic vein which moves out of the liver and it carries oxygen and nutrient poor blood. So here in this blue part there is no oxygen or less oxygen and very less nutrient. So these things are carried from the liver, and where is it carried to? It is going to the heart. So it is going to the heart for oxygenation. So hepatic vein and hepatic portal vein do not mix these two things. So for convenience, you can tell the vein which brings nutrient from the intestinal tract to the liver as portal vein, and the vein that carries blood from heart to the liver is hepatic artery. So it will not be confusing that it's an artery. So hepatic vein is blood moving from the liver to the heart for oxygenation. Now let's come to the gallbladder. The bile pigment, cholecystokinins. This is another very important term we will learn. So the bile has got bile pigments and bile salts, which we have already studied earlier. Now here comes the importance of cholecystokinin. The hormone causes to release bile by contracting the gallbladder. Now you must be wondering, the bile is coming from the liver and it is coming from the hepatic, no, this is the liver, and from the common bile duct. Common bile duct is different thing. So hepatic duct, these things come to the gallbladder. So right hepatic duct, you can see over here if I enlarge the picture, this is the right hepatic duct and the left hepatic duct, and then a common hepatic duct that comes, and then it comes and puts its content, that is bile, because bile is manufactured by the liver. It puts the bile inside the gallbladder, and this gallbladder keeps the bile stored and ejects the bile when there is food inside the stomach. Now who makes the gallbladder to eject the food? This is the point that I was talking about. This hormone, the hormone cholecystokinin, is responsible for contraction of the gallbladder wall, and it causes, by this contraction, just like a pichkari, spray, it's sprayed inside the intestinal lumen, that is the duodenum, through the common bile duct. So absorption of bile salts and fats happen at the ileum. So at duodenum there were many interesting things happening, where the bile was acting on the duodenum, and then pancreatic lipase, pancreatic amylase, pancreatic protease, but absorption paths come to the next part of the small intestine, that is ileum. So bile salts from ileum go back to the liver. Are you aware of it? That the bile salts that was secreted by the liver and through the bile duct actually goes back to the liver again, and that happens from the ileum. Now the pancreas, the very, very important organ, I'm trying to cover in a hurry. So this is pancreas. So this pancreas, this is called the lion of the abdomen. This is called the lion of the abdomen. This sits in, it's retroperitoneal in nature. So all the contents of the abdomen is actually inside the peritoneum. And this pancreas is called a retroperitoneal organ, which is just behind the peritoneum. It has a different casing of its own, and this is the only unencapsulated organ. Everything else apart from the pancreas is protected by a capsule, the liver and the kidneys. So everything has a capsule, but pancreas is an unencapsulated organ. So if it ever comes to you as a question, name the unencapsulated organ, you can tell it's the pancreas. And it's called the lion of the abdomen because it does so many important things. So first important thing is exocrine function. We discussed this, so we'll cover very quickly. It secretes the bicarbonates, which neutralizes the acid coming from the stomach. So it secretes the bicarbonate to neutralize the acid and make it neutral. It has the amylase to break down the sugar. It has a lipase to break down the monoglycerides and diglycerides and glycerol and fatty acids into components, smaller components. So proteolytic enzyme, trypsinogen and chymotrypsinogen, which is a zymogen, as you know, they are inactive, they start acting. So these are all secreted by the pancreas. We know all these things. Now we'll come to something that we have not done earlier. That is the endocrine pancreas. The endocrine pancreas is, it releases hormones from the 3 different types of cells. So the cells that are there in the pancreas, these are called islets, and these islets, they are located in different islands. If you see a microscopic view of the pancreas, you will feel like these are all different islands. So the alpha islet cells, they secrete glucagon. So glucagon is something that breaks down glycogen into glucose. So it breaks down, and it breaks down the stored thing. So glycogen was stored, it breaks down the glycogen into glucose. Now there is something called the beta cells, and this is very, very interesting. The insulin hormone. The insulin hormone is secreted from beta cells. Sorry. So alpha cells, they secrete the hormone glucagon, which breaks down glycogen into glucose, and beta cells, they secrete insulin, which makes glucose available in the blood to form into glycogen. So that is the reason the insulin actually reduces the sugar content of the blood, because it gets converted from sugar into glycogen. So glycogen is built up for storing, and that is the reason when insulin becomes less. So there are two conditions. One is called type 1 diabetes. You know that insulin is related to diabetes. So type 1 diabetes is when insulin is not produced. So the sugar content of the blood is high, and that is called hyperglycemia. And this is because insulin is not produced. This is type 1 diabetes. And the type 2 diabetes is insulin is produced by the insulin receptors. There are receptors to receive the insulin so that can act properly. So the insulin receptors are actually broken down, and that is the reason insulin is not acting. That is called type 2 diabetes mellitus. And the third thing is called the delta cells. The delta cells contain something called somatostatin. The somatostatin is a very interesting hormone which stops, like stat or station, it means to stop. So somatostatin is a hormone that will stop action of other hormones. So what will it do? It stops the action of other hormones, and that is what the action of somatostatin is. So keep that in mind. In the large intestine, what happens? Practically no digestion takes place in the large intestine. In the colon, water is absorbed. So in the colon, if water is absorbed, second, I'm in a hurry because I have to finish the class within 5 minutes, because today we will cover digestion all over. So large intestine, which is 1.5 to 1.8 meters in length, इसका size actually small intestine से छोटा है, but इसको large intestine इसलिए बोला जाता है क्योंकि इसका lumen चौड़ा है, it's thicker. Small intestine is narrower. So practically no digestion takes place in the large intestine. In the colon, there are 3 parts of the colon, that is immediately after the third part of the small intestine. So first part is duodenum, the second part is jejunum, and the third part is ileum, where most absorption happens. So ileum comes and meets the cecum. This is called the ileocecal junction, where the appendix is located. Appendix is a vestigial organ which does not have any function according to many scientists, but while a few group of scientists, they believe the appendix is a host of good bacteria that is stored inside the appendix. The opinions do vary, but again, appendix is a vestigial organ, and it is normally operated out if the abdomen is operated for any reason. So when the cecum starts, there is the start of the colon after that. So what is the colon? The colon has got 3 parts. It starts from the right side, called ascending colon, which goes up, then comes a transverse colon, and then comes the descending colon at the left side. So colon is ascending, transverse, that is goes across, and then descends. Now what happens in these areas? Water is absorbed. So if water is not absorbed properly, what will happen? You will get diarrhea. So diarrhea is caused because water is not absorbed by the large intestine properly, and too much water is absorbed, you get constipation. So if for some reason too much water is absorbed, you get constipation. Inorganic ions are absorbed, like sodium and potassium, that is also absorbed in the large intestine. And rich sources of microorganisms. So this large intestine is a source of microorganism. A lot of microorganism is there, and the feces that we excrete is also full of microorganism. That is the reason it is said you have to have a proper toilet to pass the feces, because these microorganisms can spread if you defecate or pass stool in the open. So microorganisms are there for what? They are also for a purpose. They are assisting in digestion, because these digestion of these microorganisms, there are certain things like cellulose which cannot be broken down by the human beings. So cellulose can be broken down only by microorganisms. So these microorganisms act on substances which is not normally broken down by the human digestive system. And remember, when this microorganism acts on the carbohydrates, they produce methane and hydrogen sulfide, and that is the reason the gas that is passed after you eat or something, that smells so foul. It's mainly because of methane and hydrogen sulfide. So the gas that is released is full of methane and hydrogen sulfide. So indigestible matter becomes compacted to form feces, and feces move towards the rectum, and the rectum, it's stored over there for some time, and then the two sphincters, that is the anal sphincter has got an internal one and an external one. So internal anal sphincter is formed of smooth muscles, and it is controlled by the autonomous nervous system, and the external sphincter is controlled by the skeletal nervous system, and that is under our control. The autonomous nervous system is not in our control, so the internal sphincter is not in our control, but the external sphincter is in our control. So that is the reason you can pass the stool whenever you can or you want to. And that is sometimes when you get diarrhea, the internal sphincter, along with the internal sphincter, the external sphincter also does not function properly. So unknowingly, stool is passed even without the knowledge of the person. And the external sphincter, when it becomes very, very old, in old people, the stool is passed without their knowledge, because the external anal sphincter becomes very, very loose. So this is the last slide, and after that I have this class for the next group, that is 1 and 2. They are waiting for the quiz. So control of the GI tract. Now you understand this thing very clearly. The control of the GI tract is very, very important. It has a different nervous system. The GI tract has a different nervous system called enteric nervous system. That means what? That means the nervous system can function without sending message to the brain and the spinal cord to regulate its action. So understand this. This is very, very important, guys. The intestine has its own different type of nervous system called enteric nervous system. Now how do we know it? For example, if we have some awesome food, very tasty food, we start something called gastrocolic reflex. What is this gastrocolic reflex? What it does? First, there are this gastrin that is released from the mucosal cells. So when gastrin is released from the mucosal cells under the action of these nervous system called enteric nervous system, there is food in the stomach. The stomach nervous system senses it, and gastrin is released, and it tells to release the hydrochloric acid. So hydrochloric acid is released by the stomach under the action of gastrocolic reflex, and so is pepsinogen, and it increases the stomach motility. So chyme formation, the stomach movement starts happening faster under the action of this hormone called gastrin, which is stimulated by the nervous system called gastrocolic reflex. The next thing is the pH of the stomach is 3. When the pH of the stomach rises to 3, that is, normally hydrochloric acid has a pH of 1 and the stomach pH is 2, but whenever it goes to 3, it decreases the gastrin release. So the moment it goes to 3, the gastrin release is stopped. That means what? That means the food is not there in the stomach anymore, so no need of secreting hydrochloric acid. And in the duodenum, there's another hormone that takes care, that's called the secretin. The secretin releases the pancreatic hormones and pancreatic enzymes and the bicarbonate rich solution from the pancreatic juice. So secretin, it's not creatine, it's actually secretin. It is actually secretin which releases which causes the pancreas to release bicarbonate and pancreatic juice. So the moment the secretin is released, the gastrin is actually stopping. So gastrin stops producing hydrochloric acid, pepsinogen, and the function is taken over under the action of secretin by the pancreas and pancreatic enzymes. So secretin, and the secretin is also not only in the duodenum, it's not acting only in the duodenum, it's pushed back into the stomach to inhibit the acid reflex. And cholecystokinin, it's a big name, cholecystokinin is released from the intestinal mucosa, and it acts on the pancreas and enzymes and the gallbladder to contract. So cholecystokinin is the hormone that will cause the gallbladder to contract, and it will push in more bile into the digestive tract. So now we have covered the entire digestive tract. We know what's happening in the mouth, we know what's happening in the esophagus, we know what's happening in the stomach, we know what's happening in the small intestine, duodenum, we know what's happening in the ileum, jejunum, and then it comes to the colon, what's happening in the colon, and ascending colon, transverse colon, descending colon, all the absorption, and how these different enzymes are released under the action of a different control system called gastrocolic reflex. So this is very, very important, guys. If you have any questions, I cannot prolong my class any longer today. We'll catch up with a new topic tomorrow. So till that time, bye-bye. And if you have any questions, do not forget to ping me on my WhatsApp number. I will definitely give you the answer later on. So thank you. Getting ready for the next class. Bye-bye.